Hello, everybody. My name is Juan de Bernardi. I'm the manager of the UC Davis Plant Transformation Facility. And in this presentation, I will talk about the work we have been doing in collaboration with my colleague, David Tricoli. And this project is being supported by the CDFA Beer Disease Board. Okay, the main goal of this project is to contribute to develop resilient, high quality grape cultivars, which will include cultivar with improved flavor, larger berry size, berry color, increased cluster size, but also plants that are more resilient to the environment. And that can include, for example, tolerance to droughts or salt or high temperature. And in particular, in this project, we are focusing or very interested on developing disease-resistant cultivars. We have traditional methods to improve cultivars, for example, those that include bringing genetic diversity by crossbreeding, that is crossing two cultivars with the desired, one with the desired trait that we want to bring, and them having that in the progeny, or using chemical mutagenesis to produce novel genetic modifications. However, these techniques have limitations. For example, crossbreeding is successfully used to generate improved table grape cultivar. However, if we use crossbreeding for wine cultivars, the crossing will destroy the fidelity of the clonal cultivar. So in the case of clonal cultivar, where we need to maintain the fidelity of the Sherwood plants, we need different technologies. And we are lucky that we have those technologies now on hand, like transgenesis or CRISPR or genome editing breeding. These two technologies are very powerful because we can introduce genetic variation into the cultivar without affecting the clonal genotype. But in the case of transgenesis, we have again problems because there is this approach, even when it's very powerful, it still have limitation because of public acceptance. So, however, in the case of CRISPR or genome editing, this is a very interesting alternative. It's a technology that was developed a few years ago. And it's very interesting because with this technology, we can introduce a specific modification into the plant genome without the requirement of making transgenic plants. We can imagine CRISPR or genome editing as molecular scissors. So those molecular scissors are basically a complex of a protein with another molecule that is called guide RNA. And that scissor we can somehow design and introduce into the plant cells to produce cuts in the DNA at a very specific position. So we can design this scissor to be very specific and to introduce cut at a very specific position in the genome. Once the molecular scissors, CRISPR, introduce double-strand breaks into the DNA, the cell will try to repair that double-strand break. And in that process, different things can happen. So the DNA can be repaired without any error. That's the more likely process. But there are two alternative pathways that can happen. One is that after the cell attempt to repair the DNA double-strand break, it will produce mutations or errors that will, for example, disrupt or knock the gene function. So that's very interesting because using that pathway, we can turn off the activity of certain genes. Another possibility, for example, is that if in the plant cell there are DNA that can act as a donor DNA, the cell can use that to integrate and repair the double-strand break, but in that process will introduce DNA at very specific position. And how can we use CRISPR on grapes? So the first thing that we need to do is to introduce the CRISPR scissors into the plant cell. But this is not an easy task since the plant cells are covered by a very hard structure that is called the cell wall. So the cell wall will prevent the molecular scissor to go into the cell. So in order to get into the cell, we first need to remove the cell wall and produce cells that are wall-free and are called protoplasts. Here, for example, I'm showing a solution of protoplasts. These were generated by treating the plant cell with enzyme that will degrade the cell wall. So now we have individual cells without the cell wall. Those cells can now be easily transfected with the CRISPR scissors that can go into the cell and cut the DNA at a very specific position. However, these are single cells without cell wall, and you can imagine that the next challenge now is to go back from here into a whole plant. Going from protoplast to whole plant is a very difficult process. It's very challenging, but we are lucky because my colleague, David Tricoli, with funding from CDFA, he was able to develop a very efficient method that allowed us to generate first protoplasts, introduce the CRISPR scissor into them, and generate whole new plant from those protoplasts. That is an amazing protocol, an amazing platform that has allowed us to start generating germplasm in grape cultivar to improve different traits already. Particularly in this first round of germplasm development, we have made use of this CRISPR output that allow us specifically to turn off the activity of different genes. And we have focused on 4 different traits. So one of them is flavor, seedless, browning, and disease resistant, specifically powdery mildew resistant. And we can do that because we know that in activating the activity of different genes, we can produce the trait that we want. For example, in the case of flavor, we know that in activating the genes that produce the enzymes OMT, we can reduce the green pepper flavor in the fruits. We also know that in activating the activity of genes that produce the PPO enzymes, we can reduce oxidation or browning in the fruit. Moreover, and this is a very interesting case, we know that we can, by inactivating the genes that are called MLO, we can confer powdery mildew resistant. That is because the fungus require the activity of those genes during the infection. So if we can inactivate them, now the fungus cannot infect the plant, and that is the reason the plant became resistant. We also target HEL11, and that gene, for example, we know that when the activity of the gene is disrupted, it results in seedless fruit. So the other thing that is very interesting or important on the platform that we have developed and we are working on is that we not only that we focus on 4 different traits, but we can introduce this trait in multiple cultivars. For example, I'm showing here plants from Chardonnay, Columbar, Merlot, and Cab Franc, and also from the table grape tops and seedless that are growing in our installations. And all those lines have been edited or modifying the trait that I mentioned. We have currently them in our facility, and those will be available for industry and collaborators. The current project that is being funded by CDFA, we decided to expand the capability of our genome platform in BTs to incorporate also the other pathway that is the what is called knock-in or gene insertion. So in that pathway, as I mentioned before, during the transfection in the protoplast, we can integrate or we can incorporate donor DNA for a precise insertion in the genome of the plant. And incorporating this technology into our platform will be very important because it will allow us to incorporate genes from grape germplasm collections or different wild relatives that can confer important agronomic trait without the need of doing breeding crosses that will destroy the clonal germplasm. One important example, which is an objective in this proposal, is to breed from into Vitis vinifera cultivar genes from Vitis arizonica, a wild relative that confer resistance to Xylella, the bacteria responsible for the Pierce disease. So, but first, to have this platform, this technology working in our hands, we have to test different technicals and parameters that include, for example, how to deliver into the protoplast the CRISPR scissors together with the DNA that will be a donor and integrating into the genome. So There are different options to do that, and we had to test them. For example, it could be DNA, or it could be DNA with chemical modification that will make it more stable and more efficient to integrate into the genome. So we have been doing different and several experiments where we can affect protoplasts with a mix that include the molecular scissor with the donor DNA. The idea is that here the molecular scissor will cut at the specific positions, and then the donor DNA will be inserted. So we have regenerated plant from those transfected protoplasts in two different cultivars. For example, here is a Columbine and Chardonnay. Then we have been doing a molecular analysis, a genetic analysis to determine if we were successful to insert DNA at a specific position. So the result of that we have obtained so far confirm that yes, we have been successful on integration of DNA donors into the specific loci on the grape gene. We have also observed that chemical modified DNA, the one that I say that will make it more stable, improve integration. And we have also been able to regenerate whole plants from the transfected protoplast and confirm that they contain the integrated DNA. This picture, I'm showing you the different regenerating plants that were regenerated after the transfection of the mix with the donor DNA. And all these plants contain the fragment that we wanted to knock in. And what I'm showing here is that, very importantly, we were able to demonstrate that some of those plants contain the whole fragment. So, and the size of this whole fragment, that is very important because we want to integrate whole genes, not just pieces of genes that we can obtain from different sources. So we need to incorporate whole genes, and those whole genes have a specific size that need to be integrated. In those experiments, we were able to recover plants that have fragment of the size that is expected for whole genes. So now that we have a platform and a technology that it seems to be very efficient to perform knock-ins, we want to use it actually to develop products or germplasms that can be used to improve grapes. As I said before, one objective of our project was to incorporate Pierce disease resistance into these vinifera cultivars using this platform. So to do that, we are working in collaboration with Dr. Dario Cantu at UC Davis, whose group has identified 4 candidate genes in Vitis arizonica that can be involved or mediate the PD-resistant genes. So we have now 4 candidate genes that we have transformed them into donor DNA, and we are performing knock-in experiments to integrate them into the genome of wine and table grape cultivars. So the goal will be by the end of this project to have plants with knock-in event of this potential gene from Vitis arizonica and evaluate them for PD resistance. With that, I would like to thank all the members of the plant transformation facility, specifically David. David was fundamental in establishing the protoplast platform at our facility. And he is still very involved in doing all the protoplast work for this project. I would like also to thank Lucero and Mariana that are staff in our facility and provide a lot of support for the genetic and molecular analysis of this plant. I would like to thank also Dario because he's a great collaborator that provides all the sequences, genome sequence from different grape cultivars and the candidate genes for Pierce disease. And finally, I would like to thank CDFA for the support, financial support to perform all this work.